Inga mana, inga reo, inga tā wira pumanawa, ro rakatira ma, tena koto, tena tato katoa. Nō reira, ko waio, ko Harleen Hain, tako ingawa, ko te tumuaki o te whariwanaka, a tako, aho. Nō reira, ngā mihi nui, tena koto, tena tato katoa. My name is Harleen Hain, and I have the great privilege of being the Vice Chancellor here at the University of Otago, and it is my great pleasure to welcome all of you to this inaugural professorial lecture for Professor Peter Fineran. On behalf of the university, I would like to give a very, very warm welcome to the members of Peter's family who have joined us today for this fantastic event. His dad and stepmom, Brian and Barbara, are here. Welcome to you, and his partner, Laura. It's fantastic to have you here, and I know it's very important to Peter to have you in the audience for this very special occasion. In addition to these very special guests, as I look out through the audience, I see lots and lots of very familiar faces here. There are academic staff, professional staff, and students from around the university. I'd like to extend a particularly warm welcome to our Chancellor, Dr. Royden Somerville, who is up at the top back there. To all of you, no mai, how mai, welcome. Now, these IPLs are an incredibly special time for us as a university community. We have all come together tonight to share not only in Peter's success, but also to learn from him. Professor Fineran is a creative, prolific, and very well-respected scientist. He is a gifted and popular teacher at both undergraduate and graduate level, and I suspect that a large number of his past and present students are in the audience this evening. Last but certainly not least, he is also a genuinely nice guy who has provided exemplary service to his department, to the university, and to his discipline more generally. This triple crown, outstanding research, teaching, and service, is the true hallmark of a University of Otago professor. So Peter, on behalf of the University of Otago, I would like to congratulate you on your very well-deserved promotion to professor. I will now call on Professor Vernon Ward, who is the Dean of the Dunedin School of Biomedical Sciences, to tell us just a little bit more about Peter's academic journey to professor. Nō reira, tena koto, tena tato katoa.
Thank you, Vice Chancellor. Tena koto katoa, hame nui tere kia koto. Welcome, everybody. This is Peter Fineran's inaugural professorial lecture. And so an IPL is a very important occasion. So thank you very much for coming on to celebrate this. It's great seeing a full hall of people here and to see so many microbiologists in one place. It doesn't happen very often. It's my role to give you some background information on Professor Fineran's career, and it's been an incredibly strong career, and it's not possible in the 5 minutes I've got to cover everything that he's done. So I'm going to give you a snapshot and then indicate the areas that are just as good that I haven't covered. He received his Bachelor of Science Honours degree in Biochemistry with first-class honours from the University of Canterbury in 2001, and he followed that with 15 months of work at the John Curtin School of Medicine at the Australian National University in Canberra. He then followed that, he got a Bright Futures Top Achiever Doctoral Scholarship, which took him to Cambridge for his PhD, and then he carried on and did a postdoctoral fellowship at Cambridge after achieving his PhD. He then came to us here at Otago in the Department of Microbiology and Immunology in 2008 as a lecturer. He became a senior lecturer in 2011, an associate professor in 2015, and now professor. And in essence, from when he would have found out about this, effectively 10 years from lecturer to professor, and that is a phenomenal path. With his research, there's a few things we do to measure research. One of the key peer assessments of your research is your ability to draw research funding. I often look at it from that perspective rather than the dollar perspective. 4 Marsden grants as a PhD, as a PI, Rutherford Discovery Fellowship, a member of the Bioprotection Centre of Research Excellence, and he's had also, in addition to a bunch of other grants and funding, he's had industry funding from the kiwi fruit industry, so he's not just an academic sitting in his ivory tower, he also gets out there in industry as well. I was looking at his supervisory record, whether it's from advisory panels, PGDPSIs, honours, postgraduates, up to PhD supervision. And the last count I have is 65 postgraduate students. And he's hosted international students on internships. And he's taken at least one summer studentship every year the entire time he's been here. So it's very vast. His distinctions that go with this are numerous. I've already mentioned his Bright Futures doctoral scholarship and his Rutherford Discovery Fellowship. He placed third in all of the UK in Young Microbiologists in 2006, so great start, Peter. You've done better since then. Finalist in the Prime Minister's McDiarmid Emerging Researcher Prize, the School of Medical Sciences Emerging Researcher Award, the University of Otago's Rohith Trust and Carl Smith Medal, Thermo Fisher Scientific Award for Excellence in Molecular Biology, New Zealand Society for Biochemistry and Molecular Biology Custom Science Award, the Ross Crozier Medal from the Genetics Society of Australia, the Biomedical Sciences Distinguished Researcher Award, and he's just back from receiving the Fleming Prize in the UK, which is obviously a very prestigious international award. So that's just a selection. He publishes prolifically. I had a look this morning in PubMed. There's already 7 articles this year, already in very good journals, including Nature Microbiology. It tells me there's a total of 76 publications there. And to give you an idea of the quality of them, these are in Science, Proceedings of the National Academy of Sciences, Molecular Cell, Nature Communications, Nature Microbiology, Nature Reviews, Microbiology, Nucleic Acid Research, Plostogenics, MBio, eLife. 5 of those, Web of Science tells you, are highly cited special papers. It's a phenomenal record. He gets commentaries in the journal Science, multiple Nature publications, Faculty of 1,000. He's on the media, he's in print, he's in radio, he's in television. It's a phenomenal record. I had a look, because of what he does, I had a look at, his Web of Science H-index is 31, but I noticed there's quite a few papers missing there, so it's obviously better than that. And his H-index by Google Scholar for the last 5 years alone is 33. So I don't have enough time to talk about all that communication of science that he's done. He has a very wide-ranging speaking schedule and international conferences, invited speeches and what have you, and to say in the media. You might wonder whether that research record goes with his teaching and service record. I don't have time to tell you the list. It's as long in each case, and it's outstanding. He is one of our very best teachers. He receives top evaluations of his teaching from students. The comments he receives are ones we would all be very proud to receive. His service contributions are broad, both internally to his department, to his university, nationally and internationally. So it's an incredible record. So this brings me to the point of introducing the talk for tonight. Professor Fineran will explore the resistance of bacteria to viruses. I'm a virologist, this is great stuff, right? And he'll explore the innate defences bacteria have at a population level to shut down a viral invader, as well as the hugely topical CRISPR-Cas system, which many of you will have heard of. It's a virus resistance mechanism. And he'll look at the battle between the bacteria, the host, and the virus, the invader, and the balance that they have. So with that, I'd like to invite Professor Fineran to present his inaugural professorial lecture. I wondered if this is a nod to Trekkies, but anyway, if not, resistance is not futile. Bacterial, innate and adaptive immune systems. Nō reira, tēnā te mihi kia koutou katoa. Tēnā koutou, tēnā koutou, tēnā tātou katoa.
Thank you. Kia ora koutou. Thank you very much, Harleen and Vernon, for such a nice welcome and introduction. And thank you so much for all of you for coming along tonight. It's really a pleasure to see, so many friends, colleagues, neighbours even, and of course to have my family as well here is really fantastic. So what I want to do tonight is to give you a flavour of an unseen arms race, a battle that's going on out there in nature between bacteria and the viral invaders. And before my lab blames me for forgetting to thank them, I really wanted to thank them for being the critical part of this whole process. And I think, I've been very fortunate to have an incredible group of both past and present students and staff over the last 11 years here at Otago. And Vernon said there were 65. I counted about 50 or so, I need to keep up. But they're really the people who do all the hard work, who are so passionate and really make it so much fun for me to come into work every day. But also, I've had really fantastic collaborators, and I'm not naming them all on here, but basically a lot of great people here at Otago, as well as internationally as well. So I haven't always wanted to be a scientist, but I had some very inspirational people while growing up, and my parents were both botanists, and so I clearly had some influence from them. But I also had some really good teachers, and so I had one teacher stands out at primary school, Mr. Falloon, and he was a teacher that got us to do all these hands-on things. So he would ask us to do things like take a marble and make it fall from 1 meter to the ground and see who could do that the slowest, and all these sorts of things. And that really got you thinking about science, got you coming up with ways to tackle problems. And I recently did a bit of a literature search to find out the early influences of my science career, and rather than using PubMed, I had to go through the boxes in my parents' attic. And I found this prestigious publication in the journal Social Studies back from 1987, where I state, to quote myself, the job I'd most like to do as a scientist, because you'd be famous for your inventions. I'd invent all kinds of things and also get money for the things I invent. It would also be fun making things. I would also have my very own laboratory. So a couple of those things I think have come true. Basically, I've become a scientist and I get to make and sort of invent things, I guess, in a genetic sense. I'm not sure I'm rich and famous yet, but there's still time. And I don't even have my own laboratory because I have to share it. But luckily, it's with some really, really good people. And so if my students, who I've hopefully taught to be critical, look at this, hopefully they've spotted that my handwriting has deteriorated since 1987, but my spelling hopefully has got a little bit better. So I didn't always want to be a scientist. After briefly thinking about becoming a rock star or an artist, I decided to study biochemistry with some sage advice from my dad. And I had the pleasure of working with Jack Heinemann at the University of Canterbury, and he's really a very passionate, motivated researcher. And so as an undergraduate, he was an incredible mentor to get me excited about science. And a lot has changed since I did my honours. Probably one thing that hasn't changed is that regulations and paperwork are still making doing science a bit of a challenge. Because actually during that time, that was when the Hazardous Substances and New Organisms Act actually kind of came into force. And it turned out that during that year, Canterbury and like many other places discovered that they were not appropriately regulated, and the lab got shut down. So I was partway through doing my project and we had to stop for a couple of months actually. And that actually worked out really good. So there was a silver lining, which was that when we could start back up again, Jack said, well, we're not going to be able to do the original project, which was looking at how bacteria pass genes between themselves, because there's not enough time. But let's work on these things called bacteriophages, which have become a real passion of mine since then. So most of you are probably going, or at least the non-microbiologists are going, bacteria what? What's a bacteriophage? So like many of the people in the audience, I've had probably a viral infection recently. I've had a cold. And so, we get colds, we get infected by viruses. Plants get also infected by viruses. And it turns out that basically all cellular life can get infected by viruses. And so these bacteria over here, which are these single-celled microorganisms, they also get parasitized or infected by viruses. So we call these viruses bacteriophages, which literally means, or translates from the Greek to eaters of bacteria, because they basically can infect very specifically bacteria and destroy them, essentially eating that culture. So after doing my honours, I thought I'd have a break and look around for PhD opportunities and learn some new skills. So I moved to Canberra and worked with Ian Young on sort of a protein purification and biochemistry projects. That was a really nice chance to pick up some new skills before heading over to the UK. And this is where what drove me to contact George originally was because he had been one of the researchers very influential early on in showing that bacteria can actually talk to each other. So usually you think of bacteria as single cells, but in fact they can communicate to each other. And I thought this seems like a really cool thing that bacteria are doing, and so I wanted to study that. And it turns out really that bacteria are more cultured or more sophisticated than we might think. I'm sure they're not actually discussing Verdi or Mozart and drinking glasses of wine, but certainly they are able to engage in communication. And I think this is summarized or shown quite nicely in this picture here. So this is an agar plate of bacteria. And what you have down the bottom here is bacteria that are talking to each other, and they're telling each other to start producing an antimicrobial pigment, this red pigment. And the bacteria that are at the top here, these ones, they basically can't speak, so they've been blocked in terms of their speaking, but they can still hear. And so these bacteria are basically being told by the ones down the bottom to start producing this antimicrobial. So you can see the communication happening on that plate. And this also tells us that bacteria can work together as communities and often as groups. And that's where sort of my interest in bacteriophages and this communication came together. Because what I was fortunate to work on was a bacterial immune system which protects these bacteria from viruses, and they do this in a group behavior type of way. So I want to talk a little bit more about bacteriophages because these things are so incredible. So these are just some pictures of these different bacteriophages shown here. And they are so abundant on the planet that they outnumber all other organisms basically put together. So they are sort of the dominant organism or biological entity on the planet. And what they do is they're essentially a piece of genetic material wrapped in a coat, in this case a protein coat, and they can interact with bacteria very specifically, just shown over here. And what they do is they bind to the bacteria and they inject their genetic material, and then they use that bacterial cell as a factory to produce many more viruses. And these viruses then burst out of the cell like an explosion and then spread to other cells and then infect them. And so why is this so important? Well, it turns out that if you actually estimate how many times these viruses are carrying out those infections and exploding cells, it's something like 10 trillion times every second. And I cannot actually get my head around that number. Maybe some good mathematicians in the audience can. But to put it simply, if you look in the oceans, apparently something like 40% of all bacteria in the oceans are being turned over by these viruses every day. So these viruses are having really important roles in terms of global ecosystems and turning over of nutrients. So because of that pressure, you've got all these viruses infecting bacteria. Bacteria have actually evolved defenses. They've evolved these immune systems that protect themselves from being infected. And that's sort of where my interest now has been focused on, is essentially looking at how bacteria can fight back against these viruses, how they can kind of develop their own arms or their own weapons to protect them against these viral invaders. So I then came to Otago to set up my group and was very fortunate to have both Frank Griffin and Vernon Ward as heads of department when I was setting up my group, and both were very supportive mentors. Frank was a great character to have as a head of department when I first arrived, very enthusiastic, made me have no doubts at all about moving to New Zealand. And then since then, as I said, I don't have my own laboratory, but fortunately I get to share it with two fantastic other members of the department, Clive Ronson and Greg Cook. And since having my group there, they've both been very supportive mentors, providing a lot of very good advice, sometimes not so good, but often very good. So since coming to Otago, what I've done is I've brought all of these different areas of my research interests together, and what we're really focusing on is how bacteria interact with these viruses, these bacteriophages. So how do bacteriophages infect bacteria? And how do these bacteria protect themselves through immunity against these viruses? And this also relates to this other area, which I haven't talked so much about, but is gene transfer. So as well as getting DNA coming in from these invading viruses, bacteria also passing around genes between them. And so we're very interested in this process as well, because a lot of these immune systems that work against these viruses also work against the transfer of DNA between bacteria. So why is it actually important to work in this area? Well, as I've told you, these interactions are very vast. So you're actually, they're contributing to microbial evolution, and global ecology. These genes that are being spread around are major contributors to the rise in antimicrobial resistance that we see. So gene transfer is spreading antibiotic resistance around. And what a lot of people are also interested in is using bacteriophages, because they are very specific to kill target bacteria. We can use them and take inspiration from them to develop new microbial therapies against drug-resistant bacteria. And finally, one thing which I guess is very unexpected, but most researchers would say is not unexpected, is that when you're carrying out research, things come that you don't expect. And one thing that's really come out of the research I've shown you, not specifically from my group, but from the field as a whole, is biotechnological tools that have really reshaped our ability to perform genetics. So just a little bit more background. So why would we, or how can we use these bacteriophages in these viral defense systems? So what I've told you is that if you've got a bacteriophage infecting a cell, this will then rapidly take over the cell, produce new viruses, and explode that cell. So we can take advantage of that to kill and target very specific bacteria. So one area of research that I won't talk about is we're using this as a potential therapy to treat PSA, which is an important pathogen in kiwi fruit. On the other hand, though, sometimes these viruses can be bad. So if any of you had yogurt or cheese sometime today, you've basically had a fermented dairy product. And so to get that fermentation, you need to have bacterial cultures that are growing and fermenting that product. What happens, though, is when a bacteriophage gets in there, it completely spreads through and obliterates all of those cells. And then the problem is that culture will be spoiled, and that's a big cost to the industry. So that industry has, for many years, been exploiting these immune systems within bacteria to protect these dairy cultures from viral spoilage. So sometimes it's good to be resistant to these bacteriophages, and other times you don't want to be resistant to them. Because you could imagine if you're trying to use a bacteriophage as a therapy, if cells become immune to that therapy, that's much like drug resistance emerging. So we need ways to understand these systems and try to avoid or minimize these problems. So I think one of the most sort of serendipitous things that's come out of looking at phage defense systems or phage immune systems is the discovery of enzymes or proteins that can carry out really interesting functions. So if we go back in history, 30 years or so, there was the discovery of restriction enzymes. And these are enzymes that can cut DNA, and we can use that to move pieces of DNA from one place to another. And so many drugs, for example, insulin being a prime example, has only been able to be produced at the high kind of quantity and purity within bacterial cells because we can cut that gene out and put it into bacterial cells. So this has been really important in many areas. And recently we've had this CRISPR-Cas explosion where essentially by studying these immune systems within bacteria, we now have enzymes which we can use to modify genetic material in a much more precise manner. And that is leading to revolutions within medicine and being able to understand how organisms work, and also with potential biotech applications. And this is really important. I just did a search for some recent work or some recent things in the media, and just a couple of weeks ago, there's a report saying the first US therapy trial happening with using CRISPR-Cas. So these are technologies which are going to be changing our lives in the coming years. And probably in a different note, I was also looking at New Zealand, gene editing, and looking at the most recent link, and it was the Opportunities Party saying and putting out a little video saying that gene editing is a no-brainer. So it was a fairly simplistic policy announcement, but it's still very interesting to see that politicians are thinking about this area, which is clearly important for New Zealand and for everyone else. So one thing I've also been fortunate to take advantage of is doing a research and study leave, and so I got to work with Jean van der Oost and Stan Bruins in the Netherlands, and these two groups were very instrumental in the initial stages of CRISPR-Cas research. So I'd sort of urge any academics to do take advantage of this. It's a really useful process. So what I want to do for the remainder of the time is talk about the two areas that my lab is really interested in. The first is the sort of innate defenses that bacteria have. And then the second is focusing in on what we've been doing on CRISPR-Cas immune systems. So what are these sort of innate defenses? It's a bit like our first line of defense against viral infection in terms of it doesn't have a memory. And so how does this work? If we think back to a virus infecting a bacterium, it's going to produce more viruses. They will spread from one cell to another, and then this will rapidly kill those bacteria in the population. If you imagine a population of bacteria where you have a cell which has one of these abortive infection immune systems, the cell that gets infected actually dies. And so the problem with that is that the individual dies, but the benefit comes at the population level because it dies before that virus can replicate, and you then suppress that epidemic from spreading through that population. So bad for the individual, but good for the population. And so how do we actually study these? Well, this is a plate of bacteria where all of this kind of cloudiness is bacterial cells. And what we've done is we've taken 20 of these phages and we've sort of thrown them onto this plate. And each one of these little zones that you see here is essentially where that virus has landed on a cell, has replicated, produced 100 viruses, those have infected more cells, and so on and so forth. So you've basically got like a little mini viral explosion happening in the plate, and you can then detect it. And that's useful because you can count how many viruses are there and how many are able to replicate. This is what would happen in a non-immune host. If we take a bacterium that has one of these innate immune systems, and we put 2 million phages onto exactly the same plate, what you can see is most of those viruses aren't able to replicate because the immune system is suppressing them. What you can see, though, is there are some viruses which are able to grow, and I'll come back to those. So we discovered during my postdoc in George Salmon's lab a system which provides this kind of abortive infection immunity. And the way this works is there are two components to the system. There's one which is toxic, and there's one which is an antitoxin. So what happens is if you express this toxin in the bacteria die, so you can't see any bacteria growing here. These are little colonies of bacteria. If you then also express this antitoxin, what happens is the bacteria can now grow. So they're basically being protected by this antidote or this antitoxin, and that's what's shown down here. So in the end, what we've got is we've got cells which contain a complex which is inactive, and it's sitting in the cell a bit like a mousetrap, poised, ready to kill that cell when infected by a virus. And Tim Blower, who is a postdoc or a PhD student in George's lab, he solved the structure of this, and it was quite an incredible structure. So how does this actually work? So here's a bacterial cell covered in bacteriophages, harboring this innate immune system. So when this virus infects, what they typically do is they take over the host and they repurpose host things for their purposes. And what this results in is, in fact, destabilization of this mousetrap, I guess, so that you now have this toxic protein being released in the cell. And that toxic protein not only destroys the virus, but it also destroys the host cell, or leads to the host cell death. So in the end, that cell is actually protected. So you kind of think that seems pretty good, a good way for the cell to stop the spread of the virus. So can the virus actually get around this? There's this arms race actually going on between the viruses and the host. And how do we find these viruses which can basically avoid the immune system within bacteria? If we come back to this picture of the plate I showed you before, remember we've put 2 million viruses on this plate, and there are some of these little viral explosions happening. So if these viral explosions are happening, that means those viruses are actually able to replicate and they can ignore that immune system. So what happens if we look at those and ask, how is it avoiding the immune system? So if we take one of these and we look at what's happened, it turns out these viruses have actually stolen the antidote or the antitoxin. So they've basically taken from the immune system their own protective factor. So what we think is then happening is that when the virus infects, it's destroying parts of the cell, but because it's producing its own antidote, that's keeping this sort of toxic mousetrap complex locked in that form which is safe for the cell. And that means the virus can keep on replicating without destabilizing and shutting the cell off. So it's been able to escape that system. So that just shows you how one of the ways that bacteria can protect themselves from these viral invaders. And one of the main interests though in my lab is looking at how they use these CRISPR-Cas or these adaptive immune systems. So what these systems are, this is just a picture of a bacterial cell. And what these adaptive immune systems are is they provide bacteria with something a little bit like our own adaptive immunity. So they are resistant to viruses by acquiring a memory from that virus and then having a specific response to protect them from being infected. And the way they work is they have two parts. One is what I'd call the memory bank. It's basically storing the memories of the past invaders. And then there are these components which are important for providing the immune mechanism. And these systems are found in almost, probably half of all bacteria. So how do these systems work? Well, first, just like our immune system, the bacteria need to be immunized. They need to acquire that memory of an infection. And the way they do this is when the viral genetic material comes in, there are these components which will take a piece of this and essentially add a new memory to this memory bank. So it's storing memories of past invaders. What's kind of neat is that these memories are stored in one location, so we can know that there's sort of new memories and old memories that these bacteria have. So memories can be formed, the cells are sort of vaccinized, but then they need to be need to actually be able to provide protection against that virus. So what they need to do is they turn that memory into something that can be read, and then they take that and they use that to guide this machinery to the invader and then destroy that invader. So they're basically taking this memory and recognizing something that looks the same as that memory and then destroying it. So it's a very cool system. And when I say it's a cool system, I mean it's a really cool systems because there are just so many of these out there. If you look within all bacterial genomes or all bacteria, there's something like at least 30 different varieties or flavors of these systems. So there's incredible diversity, and this is part of this arms race that's going on between the viruses and their hosts. So what I want to do now is talk a bit about some of the work we've done to try to understand how these systems work, to look firstly at how they generate memory, how they control these systems at the right time, and then how they can elicit that defense. And then I'll come back and talk a bit about this sort of arms race that's going on. How can the viruses come back and have a chance against this immune system? So how are these memories formed? This is the stage where the virus comes in and new pieces need to be stored in this memory bank. And so what my group has done, we showed that the Cas1 and Cas2 proteins, we showed that a couple of proteins could interact to form a complex that are important for this process. And we had a very nice collaboration with Kurt Krause's lab in biochemistry, as well as Mick Nabostina and some Dutch collaborators, where we could actually observe this complex and see how it's holding these little memories before it puts them into the memory bank. And what we thought is, can we now take what we know and can we take the minimal components to show that memory formation can happen? And what we do is we take the protein or this machine, and then we add the memory and we add the memory bank, and we do this all within a test tube in the lab. And we can see that just by combining these three things, we can actually get memory formation happening. So this tells us that these are these main components of that memory formation. So the other thing we want to do is we want to see what's happening in real cells, because that's what's happening more in nature. And we do this by taking bacteria and then adding, or basically giving them a chance to acquire immunity against an invader. So we grow the bacteria and we then ask, are they acquiring new memories from this invader? And we can do this by looking at the memory bank and seeing, has it stored any new memories in there? And because each memory is being added to this memory bank in one location, we can see that the memory storage is getting bigger and bigger as more memories are being added. And what we can do is we can look at these memories by carrying out next-generation sequencing, or basically looking at these memories and seeing where do they come from within the invader. And we've had a very nice collaboration with Chris Brown's lab for a number of years, and his team's developed some tools enabling us to detect these spaces as well as their targets, or these memories and their targets. So where are these memories coming from? What can we learn about what makes something memorable? Well, it turns out that all of these memories are very restricted in their size. So if we look at what memories are acquired, they all have a very discrete size. And they are all found next to particular parts of the viral genome. So it seems that only certain sequences within the virus are actually memorable to this immune system. But one thing that I think was the most surprising and probably the coolest discovery from some of this work was that when the bacteria are acquiring these memories, they actually make mistakes. So rather than always recognizing the viral invader, they sometimes, and actually fairly frequently, acquire memories from their own genetic material. So it's a little bit like having an autoimmune disease for bacteria, because its own immune system is thinking that self is actually non-self. And by doing a few sort of tricks, we could actually show that this leads to the bacteria essentially dying. So they sometimes make mistakes, and that's self-destruct, game over for those cells. So that got us thinking, well, if these systems are actually sometimes detrimental, they're sometimes causing the sort of autoimmune effects for cells, when are these immune systems actually turned on? Because it would make sense for the immune system to be most effective when it would be beneficial for the bacteria. And we started thinking back to how would bacteria respond to a viral infection? And if you imagine a population of bacteria with a virus infecting it, that virus will very rapidly lyse the cell, spread to other neighboring cells, and then kill bacteria in that population. So if you imagine a very dense population of bacteria, that virus will very quickly spread through that population. So it's a bit like you guys in the audience. If one of you's got a cold, pity the people sitting around you, it's probably going to spread quite quickly. And it's the same for bacteria. So we thought, if bacteria are going to be vulnerable to viruses when they're in this high density state, are there going to be ways or times that they can control their immune system to increase it in these states? And that's where it comes back to the ability of bacteria to talk to each other. It turns out that bacteria, when they talk to each other, they can actually count essentially how many bacteria are present, whether there's lots of them or very few. And so they do this by sort of secreting these signals, and when there's very few bacteria, the signals are too quiet. It's like whispering. When the population gets very dense, what happens is you've got a lot of this talk going on between the bacteria. It gets very loud, they can then hear it, and what happens is they then change their behavior, they change their gene expression. So this means that as population size changes, bacteria can alter their behavior in response. So that got us thinking, would bacteria use this type of approach to increase their immunity when they would be most vulnerable to a virus spreading through a population? And the answer is yes, that's exactly how it works. So what we can do is we can take bacteria that can't talk to each other and then see how much of their immune systems are actually being produced. And we can see that their immunity is decreased when they're unable to talk to each other. And this is also true if we look at how strong their immunity is and how well they can actually acquire new memories. So these systems need to be able to talk to each other to have a very strong immune response. And so the way we think about this now is we think if you have a small number of bacteria, the immune system is less necessary, and so it's kept at a very low level. And then when the population gets very high, the bacteria talking to each other and saying, there's lots of us here, let's increase our immunity. So that's sort of taken us to understand how these systems, or when these systems are switched on, but we also want to know a bit about how they recognize the invader. And this sort of comes back to a similar picture to what I showed earlier. If you have viruses growing on a plate, this is one which has no memory of the virus. this bacterium has acquired one memory against that virus. And so you can see that those cells now are protected from that virus. And a cool experiment that we did was we basically added more and more memories to this and asked the question, what happens? And a little bit like us, as you get greater and greater recognition or memory of someone or something, you get a stronger and stronger memory. And so this gives them more protection with more memories against the virus. So I've basically told you that these systems work and provide protection against viral invaders, and it seems that the systems work pretty well. So we wanted to know, well, we still see so many viruses out there, so they must have ways of getting around these defense systems. And that's what I just want to talk a bit about now, is the ways that viruses can evade the CRISPR-Cas system and how the CRISPR-Cas system can respond back to that. So firstly, how do these phages evade that immunity? And how do we actually study this? It comes back to these same sorts of images where we see these little viral explosions on a plate, and we know that those viral explosions mean that those viruses have evaded or avoided that immune system. And then we can look at them and find out what's happening. And by doing this, we can actually discover that the viruses are avoiding the immune system by changing. They're essentially disguising themselves from that immune system. This is a little bit tedious because you have to pick individual things and look at what's happening. So what we've also done is we've been able to scale this up and actually look at 200,000 elements that are avoiding the system and ask the question, which ones avoid and how do these systems get avoided? And I won't talk about the details, but what this tells us is essentially what are the features that this immune system needs to recognize the invaders? What are the features for recognition? But the downside of that is you think, well, you can actually find these viruses that can escape or avoid the immune system. So how does this immune system then deal with those ones that have escaped? And as part of this arms race that's going on, essentially the viruses have escaped, but the bacteria are then able to come up with their own strategy to deal with them. And it turns out that what these immune systems can do is they can recognize these viruses that have escaped, although it's sort of imprecise recognition, but this leads to a very rapid new memory formation. So I guess it's a little bit like a vaccination as well. And so what happens in these cases is when the virus has first escaped, the immune system now generates a very strong new immune response against that invader. And this often results in many new memories being acquired. And so what we would see is many memories being acquired. So it's a bit like now the immune system not only remembers, the eye color, the hair color, the height, the weight of the invader. So there's a much stronger sort of response or ability for it to recognize what might be coming into the cell. And so we thought, how can these viruses now actually evade this very strong immunity? Because it would seem that having all these recognition elements would be tricky to avoid. And so we did some work to try to see how the viruses can evade this type of multiple very strong memories. And what was really surprising was we found that the only way these viruses could evade this immune system was by removing large parts of their genetic material. So basically removing all of the bits that were being recognized. And we did some nice work with Mick Bostina as well to look at whether we could see these changes in the viral structures. And we could see that when the virus was being recognized within something that measures the length of this tail, the only way it escaped was by shortening this tail and removing the region that was recognized. And we can actually measure that and see this in the lab. So I think this shows us that we can really see these evolutionary events in very rapid timescales, which I think is really exciting. And then you can even see it with your eyes when you get to this high-end imaging. So one of the problems with that type of strategy for a virus can only delete or remove so much of its genetic material. There's a point where it can't just keep removing things that are, that the immune system remembers. It has to have another strategy to evade the immune system. And one of the things that was discovered a few years ago now, that viruses can produce molecules which can suppress these immune systems within bacteria. And so these are called anti-CRISPR proteins because they stop CRISPR-Cas systems. So they're really pretty cool systems. So what happens is the virus injects its genetic material. One of these genes will produce this molecule which then can shut down the immune system. So the virus is just coming in, shutting off the immune system, and then it can replicate unimpeded by that immune system. So we've been doing a bit of work to try to understand how these systems work and also how they are regulated. So hopefully that's given you a little taste of this incredible arms race that's going on between the bacteria and the viruses. It's really kind of a game of one-upsmanship where one will do something, the other one will come back and respond, the other one has another trick up its sleeve, and it's just a continuous sort of arms race that's happening. And I think this really tells us a lot about evolutionary processes, but then this has a lot of real-world implications and applications, as hopefully I've alluded to earlier. And that really just leads me to sort of finish on this picture, which was drawn by a former lab member, to show that there's really this incredible arms race going on between these elements. And I think the question really is what is next, because there's always something else out there, and I think that makes it interesting to come into work every day to try to find it. And with that, I'd really just wanted to thank the incredible team who've done all of the work I've shown you today, and also the funders who've made everything possible. So this is not possible to do this work without funding. And in particular, I wanted to really acknowledge the Royal Society of New Zealand, particularly through the Marsden Fund and the Rutherford Discovery Fellowships, because they provided a lot of very fundamental research funds which enabled us to do a lot of this work. And I think that kind of links back nicely to the fact that CRISPR-Cas systems are providing such a useful application and a potential biotechnological tool, but we could never have found them by trying to look for them. We had to do fundamental research to be able to do that and find them. And now the potential is massive. So with that, thank you very much for your attention. It's been a pleasure.
Thank you very much indeed, Peter. Wonderful talk. In fact, I remember meeting Peter's postdoc and PhD mentor, George Salmond, at a conference in 1985, I think it was, in the UK. And George was telling me about this marvellous New Zealand PhD student that he had in the lab, the student that would make an absolutely consummate academic, the student that was a complete package in terms of his research, communication and interpersonal skills. And I think we've seen tonight just how the truth in George's words on that. And I should say it was only a couple of years after George said that to me that we managed to get Peter into the lab anyway, or into the department. So I guess Peter has emphasised in his lecture just how much science and medicine owe to those viruses that infect bacteria. I think he has really elegantly illustrated the role of curiosity-driven research, how that curiosity-driven research into the arms race between the bacteria and their viruses has led, first of all, to some fundamental understandings and new knowledge, and wonderful new knowledge, but it's also actually spawned a number of, at least two biotechnological revolutions. The genetic manipulation is part of the everyday parlance amongst the public, I guess, these days, and it's probably not going to be too many more years before CRISPR-Cas is also an everyday word in the Oxford Dictionary as well, just showing how, as Peter has emphasised in his own talk, how important this fundamental research is and how the sort of unexpected findings can lead to marvellous inventions that we could never otherwise have imagined. Okay, so listening to Peter, I guess I could, and I'm sure you as well, could only really marvel at the sophistication of these bacterial innate and adaptive immune systems. And I think you also have to marvel at the resourcefulness of those bacteriophage in fighting them and overcoming them. And I guess the final thing you really got to marvel at is what's around the corner? What are we actually going to find out next? I mean, it's such an exciting field, and there's such a lot of ongoing, as you say, it's the arm race that is going to lead to new innovations, or more and more innovations all the time, I think. So, Peter, I guess I'll just ask you people again to join me in thanking Peter for this wonderful talk. He's really emphasised his group's world-leading contributions to these rapidly advancing fields, and we really look forward to hearing the future in a few more years' time as well. So, Peter, I think we've got a little gift sitting here somewhere. Aha! If you wouldn't mind just topping up.
Thank you so much, Clive. Thank you. Yeah.